I'd like to welcome everyone to our third introductory session on the BV BRC, the Bacterial and Viral Bioinformatics Resource Center. My name is Anna Niewiadomska. I work at the J. Craig Venter Institute, and I'm a bioinformatics analyst and an outreach coordinator for IRD and Viper, and now for the BV BRC, specializing in viruses. So this, as I mentioned, is the third of our introductory series. If you've missed the first two, which were a basic introduction to the BV BRC and information for bacterial researchers, you can find the recorded sessions on YouTube on our channel, and I'll show you how to get there later. Today, as I said, we'll be talking about what's changed for people coming from the IRD, the influenza research database, and the virus pathogen resource, and what viral researchers can hope to find in this resource. Our next couple of sessions will also be talking about how to use the workspace, how to upload and share data. On the 25th, we'll be talking about how to put together a really nice data set, how to parse the data, how to upload and download data that's relevant to you. And following that, we'll start some more sessions on specific tools and services that you might be interested in using on the site. So just a quick overview of what the plan is today. I plan to just go over, present a brief, a few slides about what's new, what's changed, and then we can go over to the website where we can just kind of walk through how to find viruses, where to find the services that you need. And then we can leave some time at the end for discussion and any questions that you guys might have. So if you are not familiar with the bioinformatics resource centers, these are databases that are funded by the NIAID at the NIH to basically give researchers an easy way to explore bioinformatic data and to use bioinformatic tools to analyze bioinformatic data relating to particular pathogens called category A through C pathogens that are of particular importance to humans. So throughout the years, there have been several databases. Most recently, there have been 4 databases, or 5 if you're counting IRD and Viper, that focus on viruses. The Patrick database, which focuses on bacteria. UPathDB, which focused on eukaryotic pathogens, and VectorDB that focused on the vectors that transmit many of these pathogens. And in our latest round of funding, it was decided to merge some of these resources so that some of the different tools that were unique to one site might be available to users of both sites. So the eukaryotes and vectors were merged into one site called VUPathDB, and that's our sister BRC. And our sites, IRD and Viper, were merged with the Patrick database to make a new website called the Bacterial and Viral Bioinformatics Resource Center, which is what I'll be discussing today. So you can find our new website at bvbrc.org, and it's going to look a little bit different for you if you're coming from IRD or Viper, but we tried to maintain some of the same structure, some of the same visuals, and of course, the same data and tools that you're used to using, but with some improved functionality. So I thought I would just go over a quick, some quick questions that we've started getting from some of our users. And those include things like, one of the first things I get asked is, are we gonna have to create a new account? Are we gonna have to re-register? And the answer is no. So we've ported over your accounts and your passwords so that you can use the same login and you already have access to the site. So you don't need to create a new account. unless you're completely new to the site and you've never used Viper and IRD before. You can browse through the data on the site without logging in or without creating an account. But if you want to use the tools, you do have to, or if you want to save information, you do have to create an account, because that's where all of your jobs and analyses get saved into your own private workbench. So if you're coming from IRD and Viper, another question I get is, what will happen to the data that's already in our workspace or workbench in IRD and Viper? So unfortunately, because of the different backend structure of the database, we couldn't just port over the data. So if you have a few things or a small amount of data, it might be just easiest for you to download that and re-upload that on your own. If you have a large amount of data or specific data sets or analyses that you're very attached to, then please do get in touch with us and we will help you migrate your data. And you can do that simply by going to the help section at the top of the web page and just clicking on contact us and say, hey, you know, I need you guys to help me move my data over, and we can work on that for you. So in terms of what pathogens are here, what can you expect? What's new? Well, in addition to being able to now find information on bacteria and archaea, we've also added a few new virus families. So if you have a virus that previously wasn't in Viper or IRD that you're interested in, it's possible that you can now find it here. And I'll go over those new families that we've added in the next couple of slides. So has anything changed in terms of how you'll be able to search for your data or the types of data that you find? So short answer is no. You'll still be able to find the same types of data, you know, genes, genomes, proteins, epitope data, all of that will still be there. But we're hoping that the new website will make it easier for you to search for the data that you want, because we've added a few new search facets, and hopefully the new way of searching for your data will make it easier to kind of find exactly what you've been looking for. In terms of tools, has everything that was available in Viper and IRD moved over to BV BRC? So that is the goal. For now, the site is still in beta and it's not completely finished. So there will be a few bugs. Keep your expectations realistic. And if you do find a bug, please, I encourage you to, again, get in touch with us and let us know what problem you're having so we can fix it. But almost all of the tools that you can find in Viper and almost all the services have been ported over, and those that haven't will be by the end of the year. And in addition, you now have access to several new tools that you didn't have in IRD and Viper before. And again, I'll be introducing those. And what about changes to how we upload data, how we download data, anything else? So one of the things I'm most excited about is the new site is a lot faster. Especially given the growth of the amount of data in Viper, particularly relating to SARS-CoV-2, things had been starting to get a little bit slow in the past couple of years. So the new site is a lot faster. You have the ability to upload and download large amounts of information through our command line interface and data APIs as well. And in general, services are faster as well. So that's a big improvement that I'm really excited about. So moving on to what are the new virus families that you can expect to find in the BV BRC. So this is what you will find on the viral homepage. And it gives you a little summary of everything that's in the database relating to viruses. And right now we have just over 6 million virus genomes or genome segments. And we tried to keep a lot of the same interface so that it wouldn't be too much of a change and too much of a learning curve getting you into the new site. So if you're coming from IRD, you can find all of the information relating to influenza viruses under the Orthomyxoviridae family. And there's also a couple other ways to find information relevant to influenza, and we can go over those in the website demo. And we have added a few new virus families. So for example, the parvovirus family, which is a single-stranded DNA family and causes a lot of problems in both humans and animals. For those of you interested in hepatitis B virus work, we've added the Hepadnaviridae family. And for those of you who don't care about RNA viruses at all and love double-stranded DNA viruses, you're going to be pretty happy too, because we've added 3 new double-stranded DNA viruses, including adenoviruses, Asparviridae, which has mainly African swine fever virus, which is a virus that's kind of expanding quite quickly and is of great importance to agricultural industry, and the polyomaviruses. And then on the right over here, all the way on the right, we've got our featured viruses, and these include things that have been the cause of recent outbreaks. Hello, SARS-CoV-2. And of course, one of the new things that you'll see here are bacteriophages, which previously were on the Patrick site, but really are viruses that infect bacteria. For those of you who don't discriminate and love both viruses and bacteria. So in terms of how you search for the data, things are going to look a little bit different, but you will also be able to kind of search for your data using the same metadata fields, but you can do it live using a filter section at the top of the page. And you can search for, again, all the same metadata fields like collection year, country, host group, but we've made a few small refinements that hopefully will make it easier for you to find your data. So in Viper, you could not search below a certain taxonomy level. And in BV BRC, we've made that available for you so that you can search at the species or subspecies level. As long as something has a tax ID in the NCBI taxonomy browser, you can search for it by that tax ID. And for me, sometimes it's just easier to use that than a viral name, just because it can be so much more inclusive and you don't have to parse through figure out all the different names of the different subspecies. We've also extended our Vigor 4 annotation program and databases to include more viral families. And the goal is to eventually use it to standardize annotation of gene names and proteins in the entire database, because often when you import the data from NCBI and GenBank, each virus, even though they might have the same proteins, they have different names, and it can be frustrating to put together a data set and just have to independently figure out on your own which protein matches which, but has a different name. We've also imported all of the host name curation that went into the IRD sites and extended this to all the viruses in Viper, so that it also makes searching the metadata by host name easier, so that again, you don't have to trawl through the data yourself, parsing. Let's say if you're interested in a virus that infects pigs, you don't have to find everything that's underneath swine or sus scrofa, or the various scientific names and subbreeds. And we try to put those in a common name or a host group that makes sense just intuitively to users. For segmented viruses, for those of you who are interested in working on influenza, of course, bunyaviruses, and rotaviruses, all of which have multiple genome segments, again, it can be frustrating to put together a data set when you have to try and find out which virus has maybe 2 out of the 8 segments, and you really want all 8 for your data set. So we've made improvements to how you can visualize viral strains, which will include how many gene segments are available for each strain. And then you can kind of collect all of those individual segments by strain rather than as individual contigs or individual FASTA sequences. You will still be able to customize your metadata tables to make sure that the information that you see and that you download is relevant to you. And that's something that's really important to our users and is also enabled in the BV BRC. And then something else that we would like to add is genome quality facets. So we already have the ability to search for complete versus partial genomes, but we're also working on adding the ability to kind of pre-curate the data for you and take out any sequences that have a ton of ambiguous bases or just long stretches of indeterminate nucleotides, because especially with the huge influx of SARS-CoV-2 data, a lot of the genomes have problems in them, and just curating your data set and getting a clean set of sequences can take a lot of time. So that's coming soon. Okay, so moving on from the data to the services that are available in the BV BRC. On the left here, you see a list of the different tools and services that are available. The ones in blue are meant to support both bacterial and viral use cases. And the ones in black, some of them for now are only applicable to either viruses or bacteria. And I'll show you how to kind of make that designation on the website. The goal is eventually to make any of these tools that can be applied to both bacteria and viruses applicable to both. So for example, genome assembly or reference-based assembly or metagenomic read mapping, all those services will eventually be able to use them for viruses and bacteria. And we really want to focus on providing output that's easily understandable to our users, beautiful visuals, but most importantly, something that makes sense, something that allows you to take a large data set that's uninterpretable and have output that's easy to read, easy to understand, something that you can download and put in a paper is publication ready. So if you're using one of our tools and you find something that's hard for you to understand, or you think you can improve upon it, again, please do get in touch with us and give us your feedback, because that's how we can improve and make things better for you and for other users. So I thought next I would focus on what exactly the new tools are going to be for IRD and Viper users. Because you're probably, if you're coming from IRD and Viper, you probably are familiar with the tools that are there. And although the interface is going to be different, the general functionality is still going to be there. So we have a few new tools, and some of you might already be familiar with the tools focused on SARS-CoV-2 because these were introduced in some previous webinars over the past 2 years. And these include a SARS-CoV-2 genome assembly and annotation pipeline, as well as a page that focuses on tracking SARS-CoV-2 variants. So if you're not already familiar with these, the SARS-CoV-2 genome assembly and annotation pipeline is a service that takes your raw reads or contigs, and you can upload those either as paired end or single read libraries, or you can even upload run accession numbers from SRA. And this accepts reads from a number of different platforms and has a few different recipes that you can use depending on your preferences. Or if you're not an expert on next-gen sequencing assembly pipelines, you can just leave it to the system to figure out the automatic appropriate strategy. So this uses reference-based assembly to assemble the SARS-CoV-2 genome. It will specify the pango lineage that it belongs to. It will annotate your virus for you and will also perform a variation analysis. And you can get a nice HTML output that tells you what the SNPs are, depending on which gene you're looking at, give you a nice summary of your assembly statistics, give you a coverage plot so that you can see if there are any areas where you can expect amplicon dropouts based on your variant, give you a summary table of the variation data. And of course, it uses our Vigor4 program to annotate any of the features and mature peptides in the SARS-CoV-2 genome. So moving on to the SARS-CoV-2 variants and lineages of concern tracker. This page provides a lot of different information on the variants of concern that were previously circulating or are currently circulating. And the first page gives you an overview of what these different lineages of concern are. And you can flip through a few different tabs, which will give you more detailed information on each of the lineages. We'll track the rate of growth of the different lineages. There's a genome browser which you can use to explore different tracks and explore how the different variants might have changes that affect things such as ability of antibodies to bind to specific regions. You can use our protein structure viewer to look at where these different variations in, let's say, the spike protein are. You can use our phylogenetic tree to examine large tree with all of the different variants, representative sequences from all of the different pango lineages with the variants of concern highlighted in them. And there's a couple of other resources as well. Let's see, what else is new? We also have a new 3D protein structure viewer. So those of you coming from IRD and Viper are probably already used to having a structure viewer there, but we've changed it because, again, it was starting to get a little slow and buggy. So we've started using Molestar, which is a beautiful structure viewer, has great documentation, and generally will give you really nice, crisp images, will let you hide and view specific things, change how you want to look at the structure, highlight specific areas of your structure. You can use it to kind of measure interactions. So again, I would encourage you to kind of just explore through this because it's really nice. So we've also, another thing that's going to be new for IRD and Viper users are next-gen sequencing services. So if you have raw reads from sequences with viruses in them, there's a couple of things you can use, including FASTQ utilities. And we also have de novo-based assembly services. For now, the only reference-based assembly service that we have is for SARS-CoV-2, but we do have plans to extend reference-based assembly to other viruses of interest. And please do let us know if you have specific virus in mind that you think is more important than the others for us to get started on. So for now, you can use FASTQ utilities to perform basic QC on your reads. You can trim them. You can use the paired filter function to get rid of excess pairs of reads. You can use FastQC just to kind of give you an overview of your basic sequence quality, your read quality, and I'm sure most of you are kind of familiar with those already. And you can also, for now, use, until we have the reference-based alignment services or assembly services ready, you can use the align feature and specify a target viral genome to pull out and extract your viral reads of interest. And then you can use the de novo-based assembly to put together any reads that might be specific to the virus that you're interested in, which gives you a nice output in, again, an HTML report, shows you a bandage plot of what your assembly looks like, gives you a summary of the assembly process, again, gives you an idea of the quality of your output and length of your reads. So a lot of new features. And then for those of you interested in metagenomics, we have two new tools for you to explore. One of them is taxonomic classification, which basically allows you to input raw reads and kind of get a quick overview of what's in your metagenomic sample. And then we also have metagenomic binning, which allows you to kind of separate specific reads for different organisms based on what they are, which is also pretty cool. And so this is what the taxonomic classification service looks like. You can upload your reads, and it takes, again, paired end reads, single read libraries, or if you just want to fish around in SRA, you can just input your SRA run number and upload that. And this is based on Kraken 2, and we'll give you a nice output file. So for example, in this one, this was a nasal swab from somebody with respiratory illness, and they were able to find RSV in there. And this is what the metagenomic binning service looks like. You can upload your files, again, paired end, single read, and SRA run numbers. Upload them and just push a button, tell the service to go, and it will generate bins that have different viral reads based on which virus you're interested in looking at, and you can just click on those and assemble those specific reads or contigs.
Anna, we
Yes.
had one, this is Ron. We had one question. Is the metagenomic tool limited to viruses, or does it indicate bacteria as well? It also will.
Yes, so it has both bacterial binning and viral binning. The bacterial binning works really well. The viral binning, I will give you the caveat that it's a little bit finicky because viruses often have such low coverage in these metagenomic samples, so we're still kind of tweaking the parameters. But if you do test it out and you feel like there should be viruses in that sample and there aren't, please do submit a ticket to us because again, we are still working on adjusting the parameters so that it can accurately pull out the viruses from there. But yes, it does bacteria and viruses. And then I'm just going to run through these real quick so that we have some time to demo this site. But there are also some transcriptomic tools for those of you interested in RNA-seq analysis with your viruses and looking at effects on the host, for example, and also expression import tool that will help you look at, let's say, differences in up or down regulated genes. So this is what an example of what the RNA-seq analysis looks like in a genome browser, and we'll show you different up or down regulated genes, and same with differential expression imports, which can help you create a heat map and tease out which genes are up or down regulated. So real quick, if you want to find out more, I know there's going to be a lot to learn. It's going to be a steep learning curve for some people, and we are here to help you. So we've got some instructional videos already up on YouTube, and we're going to be uploading more for each of the services that are available. And again, of course, we have the webinars that we're recording and uploading here as well. Ron and I and Rebecca and some others have also put together a ton of documentation on how to use these. And these include quick start guides, tutorials for the different tools, and just reference documentation for everything. So if you can't find it here, then definitely go to the help button and click on contact us. And, if you want to say, ask us a question, where to find something, or just complain or report a bug, don't hesitate to get in touch with us. We want to hear from you, and that's how we can improve. And if you want to follow us on social media, these are our Twitter, Facebook handles. And we also have a subreddit where we invite you to also open up discussions with the larger community so that we can also help each other with projects and just general discussion on cool things about bacteria and viruses. And with that, see if there's any questions before I move on to the site demo. Okay. All right. So can everyone see my screen? I'm gonna guess yes, there's no complaints. All right, so this is the new website, and you can find it at bvbrc.org. And if you're coming from Viper or IRD, it's going to look a little bit different, but we try and keep the same basic idea, including the tabs with all the different information at the top, and the three basic sections, including how to search, how to analyze your data, and the workbench. So you can see that translated over here. You can click on information how to search the different types of data that you're interested in. And if you are curious about the different kinds of bioinformatic data, you can go ahead and click on all searches. And this will give you a brief description of what each of the different types of data here are. And it will also give you a link to the search page for each of these data types, and also to a quick reference guide explaining what the different fields are and what the different areas in the interface are. If you're interested in the different analytical tools that we have, you can go underneath the Analyze section and click on All Tools and Services. And again, same thing here. You've got a brief description of what the tool does. So for example, the assembly service, a link to the actual tool, quick reference guides kind of explaining the different interface features and options, and as well as a tutorial if that's available. Under the manage data section, you can find information relevant to the workspace, basically how to access it. And again, you do have to be logged in to access your workspace. And this shouldn't be a problem for Viper and IRD users. You can just use your previous account. If you're completely new to the site, you can just go ahead and create an account, and you'll be prompted anytime you want to use a service to log in or create an account. And you can find information about how you can upload data, download data. You can also use our site to share a data set that you might want to publish, or even just share with collaborators. And then there's also in this fourth section, information on how to use the command line interface, data API, and our FTP site if you're interested in doing a large-scale analysis or large-scale download of data, for example. So just go through these different tabs and show you what you can find under them. So for those of you interested in bacteria, you can find information on the bacterial pathogens here. But since you're interested, since this is meant to be for people coming from Viper and IRD, you can find your virus information underneath the viral families. And down here we have a list of our featured viruses, again, most of which have been the subject of recent outbreaks. Underneath the search bar, you can find pages that will take you to advanced search pages. So let's say you're interested in finding a specific protein and you already know exactly what you want. So you can use a keyword, you can specify the exact virus that you want, or use a taxon name or ID. You can specify the host that you want, a host group. So for example, if you just want, let's say ticks, and don't want to bother with typing in every single scientific name and merging those data sets separately together, you can just use a host group. If you do want a more specific host, you can type in the scientific name over here. If you're interested in a geographic group, let's say North America or Asia, you can use that. Or if you want to drill down into a more specific country, you can do that as well. And the same goes for the collection year. You can specify whether you want only complete genomes or whether you want everything, or if you have a specific length in mind. And you can just go ahead and click on search for that. Underneath the tools and services, again, this is a list of the different services that we provide. The ones with a B next to them are generally for bacterial analysis. However, as I mentioned, we are extending some of them to viruses as well. And those with a V, for example, are only relevant to viruses, which, for example, in this case is the SARS-CoV-2 variant tracker. And those without a B or a V next to them can be used for anything. And these are divided by the various categories, such as tools that are relevant to genomics or protein, comparative protein analysis, metagenomics, transcriptomics, and a couple of utilities tools down here. Underneath the workspace tab, there's a few things that you can find. One of them is just your workspace in general. And you have a couple of different folders in here by default, including genome groups. And these are basically your collections of genomes that you've put together and saved, feature groups, and these are usually collections of annotated features from the genomes. So let's say, for example, a specific set of proteins that you're interested in, and your experiment groups, which is the default folder for where your bioinformatic analyses are saved. Public workspaces is sometimes where we put public data sets or analyses that we use for our workshops, or if you've published something or someone else has published something in a paper, you can also find it over here. On the right side, you have a list of your jobs, which is basically any tool or service that you use will get saved over here under My Jobs. And My Genomes is a collection of the genomes that you upload to the site or save in the site. Underneath the Help tab, you can find a lot of the resources that I mentioned in the presentation, including the quick start guides, the quick references that just basically run you through what the different buttons on the pages do, the tutorials for how to use some of the services, the common tasks, a command line interface tutorial, links to our webinars, instructional videos and workshops, and most importantly, the Contact Us button, which if you want to tell us anything at all, you just press that, input a subject, and then tell us that you love the new site or you hate the new site, and don't forget to include why so that we can fix it. And then if you want to learn more about the site, the team, our publications, all of that is under the About section. So getting to the meat of it, how do you find your viruses? There's a few different ways. You can just go straight to the search bar and just input anything that you want, Zika or SARS-CoV-2, or if you're interested in ticks, and that will give you a list of a summary table of all of the different information that we have on Zika, for example, including how many genomes are available, how many proteins, specific immune epitopes, how many structures. If you want to, if you have something more specific in mind and you're only interested in proteins, you can specify proteins and go in through that. Another way to find your data, if you're not exactly sure what you want, or if you just want to browse the site, is just by clicking on the Viruses tab. And that will take you to the virus overview page. And this just gives you a summary of everything that's available in the site on viruses. And you can see that we've kind of tried to emulate the interface that you find on Viper, and that's by sorting through the Baltimore grouping of viruses, so single-stranded, positive, sense RNA, and so on and so forth. All of that is available here as well, because sometimes we're not taxonomy experts necessarily, even though we work on the viruses, and it can be hard to navigate to the viruses of interest that way. So if you are interested in influenza, if you're coming from IRD, this is what you're looking for. You want orthomyxoviridae. If you are a taxonomy expert, you can use one of these different tabs up here that kind of will show you the different types of information that are available for the viruses. So if you flip to taxonomy, you can drill down into the virus that you're interested in, let's say influenza A, and this will show you all of the different subgroupings that are available and have tax IDs associated with them. So let's say I only want H5N1 viruses. I can go ahead and click on that. And you'll notice that there's a little sidebar over here that gives you information on whatever it is that you've clicked on. So if I've clicked on H5N1 subtype, it'll show me the tax ID. It'll show me how many genomes there are underneath this tax ID. In this case, there's 30,000. And I can continue browsing for viruses through that. If you click on this button here, switch to taxonomy view, it will drill down into that specific subtype for you and allow you to kind of browse through data that's strictly relating to that one subtype. So if you go to the strains tab, this summarizes information for each individual strain. And the strains tab for now is only available for segmented viruses. And what this does is it will show you every single genome segment that's available for a specific strain name. So in this case, this influenza virus from a chicken in Indonesia in 2003 has all 8 segments available, whereas the one beneath it only has 2 segments, the hemagglutinin and neuraminidase that are available. So it's a really nice way if you're working on segmented viruses to find a data set with complete segments available. If you switch to genomes, this will show you the individual genomic segments that are available for each virus. And again, if you click on a specific line item here, the sidebar is populated with all of the metadata that's imported from GenBank so that you can view it. And you can just scroll up or down and find information on how it was isolated, where it came from, when it was isolated, and all of that is available. But you might not see it immediately here in the metadata table. So how can you customize this to only show you the things that you're interested in? And you can do that by clicking on this little black button with the plus sign up here. And that will show you all the metadata fields that you can hide or view at your pleasure. And some of these might not necessarily all be relevant to viruses. You might find that there's some things that are still only make sense for bacteria or only make sense for viruses. But again, we're working on customizing the view for each viral family. And again, you know, this site is in beta. So if you find something that doesn't make sense, please do let us know. But you can customize your views through this, as I mentioned. So let's say, for example, you want to add, let's say, the collection date, the exact collection, not the collection year only. So that will be added. And if you want to widen the table and look at a few more fields, you can use this little arrow to toggle that sidebar open and shut. And there's a few things in here in this green bar that will help you browse through this data and navigate it as well. So the eye usually will open up an information guide to kind of explain the different fields that you're looking at. If you want to select multiple segments, you can just click on the shift button and press that. And now you have a group of segments, a group of viral segments. You can click on this button that will group them together. And that's how you start saving a working set for analysis. So if I click on new group, and again, it'll save this by default to your genome groups folder, unless you specify specific folder, or you want to make one. And I'll just say influenza or flu example, and you can click add, and that's done. So down here, it will show you that a new group was created, but then how do you go about finding that? And again, you can find that by going to your workspaces and going to the genome groups folder, which is where you just saved that flu example. So what else can I show you in the time that's remaining? You can toggle through the metadata and filter what you'd like also by clicking on the filter button. So again, let's say I'm only interested in complete genomes or complete segments, and I only want the ones from the most recent years. So let's say 2016, and that will select it for me. And then one important thing that you have to remember is to click on the apply button. The apply button makes sure that your metadata selection is transferred over to the proteins tab so that you can look at only those proteins from the genomes that you selected. And we can see over here that there's about 573 proteins that are from that year associated with those genomes that we selected. So just want to make sure I leave some time for questions. So I'll move over to the protein structures tab. And what this shows you is a list of the different structures that are available, let's say for influenza viruses. You can click on those. And again, this gives you, the sidebar gives you information that's associated with that individual entry. If you're interested in looking at that specific structure, you can click on the S button that says switch to structure view, and that will open up a new window where you can hopefully examine the structure that you're interested in. And this is our beautiful new structure viewer. And again, I encourage you to play around with it because it's just so nice, and it's such a great way to understand the proteins that you're working on. The domains and motif tab contains information that's, let's see, why is this? Oh, so there's no, I think we have to clear these filters so that we can see the domains and structures. So I'm just going to go ahead and clear those, and we can open up the domains and motifs. And these are the different types of motifs that have been annotated by InterProScan. And you can look at the different descriptions here. And again, you can use your filters to search for a specific type of motif that you're interested in. If you're interested in immunology, you can flip to the epitopes tab, and this contains all of the information that's been imported from PDB, and sometimes it just takes a little time to load. And
Anna, you got a question?
Sure. Let's see.
What are the structures based on? Are they computationally derived? I believe those are all the ones that we get from PDB.
Yes. So if you look at the structures, you can see where they're coming from. So the majority of these are from X-ray crystallography experiments, but some of them are from NMR experiments, and some of them more and more these days are coming from high resolution electron microscopy experiments. And all of these structures can be found in PDB, which is where we import them from. So again, the epitopes are for people interested in immunology. And again, you can filter these based on whether you're interested in B cell epitopes, T cell epitopes, whether you're interested in a specific host or a specific protein. And it's a really nice way to get a relevant data set. And so the other two tabs that we have that are specific to influenza for now are the surveillance tab and the serology tab. But I think the way things are going with SARS-CoV-2, these might eventually be extended to that virus as well. And again, you can apply the different filters that you're interested in based on the metadata and view those that way as well. So we've got 5 minutes left. I think I'm gonna stop there and see if there's any other questions. And if not, I can talk a little bit more about some other features.
Anna, can I ask quickly, really nice, can I ask quickly, how fast is the viral assembly tool? Like how long does it take?
So if you're talking about SARS-CoV-2, it's relatively quick because it's reference-based assembly, and the majority of reads that people upload are from amplicon-based sequencing. So it's, you know, on a day when there isn't a ton of other experiments in the queue, it can be as quick as 5 minutes. It really depends on the size of your data set. For de novo based assembly, it can be, it's a little bit slower. And again, it really depends on the data set that you feed it. If you have very large, very mixed metagenomic data set, it's going to be slow. And it can take, you know, minutes to maybe a couple hours. And if it's just a few small reads that have been binned through the binning service, for example, or through the FastQC utilities, then it's a lot faster, and then it's minutes.
Cool. And does it? I'm assuming it's only it's based on short read sequencing, right? Not any long read sequences, or is that also?
I think we have.
Or maybe it's a naive question for a biologist. I don't know.
I think we might have the option for both.
Cool.
Malik, do you want to take that one? Is Malik on? Let's see.
He's watching, but I'm not sure if he'll be able to answer.
All right, so I think.
We'll get back to you on that one, Omar.
Yeah, I think.
Sorry, what was the question?
Whether de novo based assembly can do long and short reads. I think it can do both.
Yes,
yeah.
It can do both.
Yeah, we have a couple of different assembly strategies that can address both long and short reads. And if you don't know what you're inputting from SRA, you can just let the system figure it out by leaving it on auto assembly based strategies.
Cool. Yeah, very nice.
All right. So if there's no other questions, I will just remind you guys that we have a few other webinar series coming up if you want to learn more about the BRC sites. One of them is the tick-borne pathogen webinar series. One of them is already recorded, and that was presented by our sister BRC, the VU PathDB. And in a couple of weeks, I'll be presenting using the BRC and demoing how to perform comparative genomics analyses on bunyaviruses in ticks. So, you know, I'd encourage you to register for those if you're interested in them, or if you're just interested in comparative genomics functionalities on the new sites. And if you want to learn more about the BV BRC and the different tools, how to use the workspace, we're having introductory webinar series every Friday for the next couple of months. And, you know, we're thinking of future webinar topics, so we're happy to take suggestions. We'll probably have another webinar specifically for influenza data and tools in the BV BRC, another one that's focused specifically on coronaviruses and SARS-CoV-2. I'm sure there's people who are also interested in how to use the command line interface and data API, and any other suggestions that you guys might have